Probably another person that doesn't need too much of an introduction. Rob speaks every year, but Dr. Rob Currie is the professor and head of the Department of Entomology at the University of Manitoba. And fun fact, Rob is actually celebrating his 30th year as a professor this year. I'm pretty sure that's accurate. So Dr. Curry's done a large amount of research over his 30 years at the university. Major focus of his research has been on how parasites and diseases and other stressors such as nutrition interact to affect the winter survival of honeybees. And today, Dr. Currie will be presenting on the integrated management of Varroa and its interacting stressors to enhance winter survival of honeybees. So take it away, Rob.
Thanks for making me feel old, Derek. As Derek said, I'm going to be talking about what I think Ian Stepler described as a title that was a mouthful at the BABA meeting earlier this week. And normally my strategy is when Daryl asks me for a title for my talk, I delay, and I usually end up being titled as the U of M research report. So this year they got a little more aggressive, and they took the title off my grant and put it in there. But the grant is, I think, essentially what they mainly want to have me talk about, and it's kind of related to new funding that you guys are committing to and have committed to, and it's looking at kind of over the next couple of years. What we could do to perhaps improve the function of oxalic acid and the way we use it and the way it works. And secondly, what we can do to continue on some of the work that you've heard me report on in the past related to manipulating ventilation within wintering buildings to control Varroa. So with that, I think as a brief introduction, I'm going to try sharing my screens, and I should warn you ahead of time that this is a talk that is going to require some action and activity on your report. I have included some polls in the talk, and Brad, my trusty assistant, has agreed to call up the polls when I ask for them, and I'm going to be asking you some questions. And basically, what I'm trying to get a feel for is what people are doing and what they would like to see done in terms of the research. We are kind of in the process of designing some of the experiments for the year going forward, and input as to what kinds of things you would like to see done would be valuable. I obviously can't do everything, and we obviously have plans that we've put in the grant that we're kind of committed to, but I would definitely like to at least get some feedback from you, and hopefully make the talk a little bit more interactive than they often are in this kind of one-way format for Zoom. So I'm going to try and share my screen if I have the capacity. And it's asking me to sign in. I'm going to ignore that. So hopefully you can see the title. Can you guys see a big green cursor? Okay, great. That'll be better than I think the other cursor that I usually use. So essentially the talk is, as I said, that horrible title, but what it really should be titled perhaps is what I know, what I think I know, and what I don't know about oxalic Varroa and winter management. And I've kind of gone through a lot of the work that we've done kind of over the last 30 years, as Eric said, related to oxalic acid, and kind of seen what we've done and maybe what we can do, where the gaps are, and what we might be able to learn going forward. On my screen, the sort of panel on the right where the speakers are is hiding some of the sources that are providing funding for this, but Manitoba Beekeepers Association, you guys are providing a good chunk of cash in order to support this. We received a small grant from Bee Made Honey to support it. We have funding from the Canadian Agricultural Partnerships, the CAP program, that matches that money. There is a sort of a Genome Canada BCSI icon there, because some of the work that we're doing with BCI is also looking at oxalic acid, and we will be doing some trials with oxalic acid, so that'll hopefully feed into AMRO's genetic and molecular markers component. And the University of Manitoba is actually, my lab is coming up with a lot of the cash that's actually supporting it. I think my my lab might be the majority supporter, and it's basically honey money that we're using that we've accumulated over the years. So we're kind of getting that honey money match to put it into research to hopefully do something that's going to be of benefit to you guys. So I hope that we get some really interesting results that come as a result of the study, and I kind of look forward to getting input from you guys as to kind of what we might want to do going forward. So, you know, we all know that one of the big problems we have here in Manitoba associated with maintaining bees and keeping them alive is tied into the concept of how to get through the winter. We do have problems managing them in the spring, and we do research kind of on that, but the main focus of this particular project is going to be on the back end of the beekeeping season, looking at what we can do to improve the survival of bees going through winter. Some of us winter indoors, some of us winter outdoors, and depending upon what we do in relation to the management, it may have differential impacts on survival of the bees, how they perform, how the colonies build up, and how they kind of function throughout the winter period. So in this particular study, part of the design is arranged to look at the impacts of treatments and then how the colonies would perform either outdoors or indoors, kind of looking at a parallel study to hopefully get some interesting perspectives on it. The main focus of both of these projects is Varroa mite. From the list that Amro had that he showed in his presentation, you probably saw that Varroa is a major factor. And when you look at things like queen loss and unknown factors, you know, the interaction between Varroa and viruses, it can be integrated in to explain a lot of those stressors that are causing problems with bees, and is often sort of linked to those other stressors, so that, you know, if you've got the one stressor alone, it's maybe not a problem, but if you've got Varroa and the other stressor, it can be a problem. So what we're hoping to do, I guess, is focus on this project on Varroa, which we believe to be probably the most serious and critical economic stressor that's affecting our bees here in the province. Varroa can be controlled in a number of different ways, and Riel was presenting on the beekeeping course on Wednesday night, and he had this list of Varroa control measures, and I was kind of looking at it, I thought, oh, that's a pretty good slide to use it. And I'll just take Riel's slide, and I'll highlight in blue the things that we've actually worked on and done controlled studies, and I was surprised to find that we've done controlled studies on pretty much everything on that list over the years. But the focus of the talk today is probably going to be, well, it is for sure going to be mostly on oxalic acid. We have been doing some work on sort of the combination of oxalic acid with amitraz as well. And we did some studies on that I presented on, I think, last year. We've got some projects that were set up last fall that are carried through into the spring that we assessed in the spring, but we're still analyzing some of the virus data from that, still processing samples in the lab. So I'm not going to be reporting on that particular one today. I'm going to be focusing on kind of the oxalic acid issues. And the other thing that's not really kind of on this list is the use of different wintering management methods, indoor versus outdoor wintering, and how they impact on Varroa. So my first sort of foray into trying to wake everybody up is to kind of get a feeling for when everybody is applying oxalic acid. Certainly the focus of this talk is essentially on when do we apply it in the fall. But some people do apply it in the spring. I kind of have heard of people that sometimes go into their winter packs and kind of shoot it in there. So there is a poll here that Brad is going to load, and the poll questions are kind of listed there. Basically, what I would like you to do is you can select as many options as you want, but the question is, I usually apply oxalic acid in the following months. So you just go down and click the particular months that you do apply oxalic acid in, and we'll get a kind of a idea of where people are usually applying it. And from what I can see with the poll running now, and people are kind of applying probably October is winning the race. I guess you guys don't get to see the poll result yet. I'm not sure if it's live or not for you guys. But I think probably that gives a pretty good idea of what is going on in relation to the results. I don't know if I have control over this, Brad, or if it's, I think he's sharing the poll results now. So you can see that the winner is pretty much October. So most of you are applying it in October. That makes sense. I think October is a month when most of the brood has kind of disappeared from the colony. Certainly when you get into mid-October, there's very little brooding occurring. There's often fairly desirable temperatures and, you know, maybe a good time to apply oxalic acid, maybe the best time to apply oxalic acid. There are a few people that are applying it in April, May, presumably trying to get those bees going out of winter before brooding starts and maybe get in a good application before the mites start disappearing from the bees and going into the brood where we know oxalic acid is not effective. A good number applying it in November and a very small number attempting to apply it in winter, probably using, you know, some sort of oxalic acid vaporizer. So it kind of looks like October is the main time that people are applying it, and probably a result of, you know, that combination of brood and the availability of good temperatures. Now this slide is not advancing for me. I'm just trying to. So if I kind of look at times when I've actually done it, these would be my answers. Quite a few of the options are on there. The ones July and August I'm not doing as a beekeeper, but I'm doing kind of experimentally. Often when designing experiments, we do them in the summer, but we don't have honey supers on the hives. Some of the new oxalic acid formulations that are being considered are being considered for registration kind of in summer when the supers are on, things like the kind of paper towel method promoted by Randy Oliver. But for the most part, you know, the registered treatments would either happen in early April or October, as most of you selected. This shows some work that kind of looks at the efficacy of oxalic acid and when it does work in the spring. And it is really effective at a wide range of different mite concentrations. These experiments that you see here were experiments I did with Paul Kozak back just after he finished his master's degree. He worked for me for a period of time before moving on to go to Cornell University and then ultimately accept his position as provincial apiarist in Ontario. And with Paul, we looked at what we could do to fumigate packages of bees as a way of controlling Varroa. And we set up packages of bees using the kind of standard American package, and we exposed them to different concentrations of oxalic acid, and we looked at different mite infestation levels in these packages. The two sort of pictures you see here are ones where we exposed them in packages with Varroa that had an average infestation of 2.5%. The one over here where the label is off my screen was an average infestation of 8%. And we had two doses. We have a black line is the control, where you can see there was no control of Varroa with no application of oxalic, not a big surprise there. We had a recommended dose, which actually was 1 gram per hive. And then we had a high dose, which was 2 grams per hive. And you can see that whether you used 1 or 2 grams per package with no brood in it before they're actually installed in the equipment, you got super good control, pretty much equivalent whether you're using the high dose or a low dose. Similarly, at higher infestations, really good control, almost exactly equivalent whether you're using 1 gram or 2 grams. And if you have super bad population of Varroa, 20%, hopefully none of you ever have populations like that, it still knocks them down really well and really rapidly and is a very effective treatment. It doesn't get them down to 0, but it does get them down to a very, very low level. So this sort of vaporization method of oxalic acid where you sublimate the material is very effective at controlling and killing the mites on adult bees at temperatures that would be kind of normal temperatures for summer in the range of about 20 degrees Celsius or so. But oxalic doesn't always work that effectively. This sort of panel that you see on the left here are some experiments we did with Rasul Bahraini back when he was a graduate student. And we didn't get spectacular results. We did see a control of mites in that particular experiment. These were applied just before they went into the wintering building. But although we saw some level of suppression of Varroa, some reduction in the percentage infestation of mites that was on those bees, we didn't actually get it down even to 1%. It was kind of, I think those mite levels going into winter were around 8%. And I think we got them down to maybe about half of that, so about 4%. And in fact, the stock that we were testing in that particular experiment, which was bred by the Manitoba Queen Breeders Association, did better and did well, whether they were treated with oxalic or not. So the oxalic happened, it happened in late fall. Conditions were pretty good in terms of the temperature we applied it at, but it didn't have a really spectacular result. The graph over on the right here is an experiment that we did with Graham Parsons while he was doing his master's in my lab, working from province of Saskatchewan. And he applied oxalic acid, and I hopefully you can see it. I can't really see it on my screen because it's hidden behind the sort of pictures of people. Maybe if I suppress the or change the view, I could probably see it, but I can describe it at any rate. The oxalic acid that you're seeing there in the green bars over on the right-hand side of the graph shows the first bar as being before basically oxalic acid was applied in early September, and after, and you can see virtually absolutely no control. It appears like oxalic acid is growing mites. And this was not a vaporizer treatment. This was a trickle treatment applied under O1, which would be early September, O2, which would be kind of a late September, and O3 would be going into October. So it was kind of like an early, mid, and late fall application of oxalic, but you can see even the late fall oxalic application didn't really do anything in terms of really knocking down those mites. Not a really impressive knockdown like we were getting in those package trials I showed you earlier. So there are a number of different ways of applying oxalic acid. Some of them are indicated in pictures on the right that would be considered vaporization methods, where you're applying it by putting a little bit of dry oxalic into a heating device that then causes it to vaporize or sublimate. And when it does that, it basically creates a dust of oxalic that goes throughout the hive. It's not vaporizing like acid, like formic acid does. Other methods include the trickle method, like the one where we applied it with Graham's research, and it didn't really work all that effectively. In that method, you put oxalic acid in a solution of sugar syrup, usually about 3.5% oxalic, and you literally trickle it in between the frames. With that method, in order to get good efficacy, it's important to put it in each box. If you have 2 boxes per hive, we did follow those directions, but didn't get great results. And the one in the middle is a treatment by Randy Oliver, which he gave a presentation on a few years ago, I think, at our convention, where he's looking into kind of a paper towel method where you mix up glycerin and put that on there. That particular method was initiated by somebody in Argentina. There are others looking at it. The tech transfer program in Ontario has been looking at it and trying to get it registered in Canada. So my poll question here for time to wake up again, pay attention, is which of these methods are you kind of interested in finding out more about, and maybe which would you like to use, which ones do you use, or which one would you like to use? And I'm kind of interested in where people really would like to kind of have most of the focus on in terms of research. And we're getting quite a range of responses coming in, which is great. It kind of looks like the 2 winners, unless my talking influences the results, are going to end up being the oxalic acid vapor treatment, which is of greatest interest, with the second kind of focus on the shop towel method, which is great to know. I think the oxalic acid vapor treatment is probably, if I could divide that bar up and say who's who. Probably would be more applicable to people that have large numbers of colonies, commercial beekeepers. For a hobbyist that has 1 or 2 colonies, the devices are quite expensive and might not be as preferable as a treatment. So the oxalic trickle or paper towel might be more suited to a hobbyist, whereas the vaporization method might be more suited to a commercial producer. So I'm going to stop sharing those results. I assume you guys can see them. Bev, can you nod up and down if you can see it? I can see you as one of the few people in there. Bev Clark, you awake, Bev? He's awake. Can you see the poll, Bev? Just nod, say yes. Yes, Derek's saying yes. Okay, great. I think I'm going through individual responses or something. So a lot of people are interested in the vapor, which is good because that's one of the things that we really wanted to focus on. So I'm happy to see that for sure. There are a whole bunch of different devices that are out there. And as many of you may know, Derek Mickelson has been hired as the lead for the Knowledge Research Technology Transfer Program. And I'm quite excited by this. We have found space for him. The dean of the university has agreed to provide office space for Derek at the university. And I think there's good opportunities for collaboration between my lab at the university and the program, and hopefully future leaders of both of those sort of institutions. One of the things that Derek got together and sent me by email yesterday was a list of some of the ones that are out there. If you look at these vaporization devices, there's the ProVap 110, the Verox vaporizer, the Varroa blaster, the Varroa cannon, VM eliminator, Scott Bee Farm oxalic acid vaporizer, multiple versions of that, Varroa cleaner, Varroa Morris durable oxalic acid vaporizer, Blythewood Bee Company. And what we're kind of interested in doing is Derek may be involved in testing some of these throughout the next couple of years. Don't want to commit him to anything, but we have had conversations about this, and I'm not going to ask a poll for what people are looking at here. But if you're using something that's not on this list, what Derek and I are kind of interested in finding out is if there's a lot of other things that are out there that aren't on this list, just put them in chat, just so we have a kind of a list of what people might have in their position, what kind of things they might be using. One device that we did, I like to always ramp things up to a ridiculous level, I guess. And one of my favorite shows on TV used to be a show with Tim the Toolman Taylor, where he'd take a very ordinary device and soup it up and make it into something crazy. As part of this experiment we did with Paul Kozak, we were trying to figure out if we could scale up oxalic acid fumigation to basically put packages in a room and fumigate an entire room at the same time. And on the right-hand side, you can see, or left-hand top corner here, you can see we're just loading bees into these packages here. So you get a large package. What you see here is a frying pan full of oxalic acid. We built a huge plastic enclosure. We basically heated up that frying pan of oxalic acid, and it's crazy. If you look, all this white stuff here, that's not snow, that's oxalic acid. So it generates this huge amount of dust. The dust goes anywhere. If you've ever done drywall in your house and you sanded the walls and you get that dust all over the place, or if you've ever been dumb enough like I have to use a cement saw in your basement, you'll find that dust upstairs in your house. And oxalic acid is very similar. It creates this fantastic dust that just goes everywhere, and then it condenses on the bees, it condenses on all the parts of the hive, pretty much everywhere. And that's kind of the mechanism of the way it works. One of the questions that we're kind of wondering about is what is the minimum temperature at which oxalic acid can be effective? And I guess what I'm wondering is what you guys think and maybe what experience you've had. So there's another poll here, Brad, for the minimum temperature at which you think it could be effective. Those original experiments we did were done in the range of about 15 to 20 degrees Celsius. Do you think it would work at 10 to 15, 5 to 10, 0 to 5, minus to 5 to 0, or below -5? What kind of range do you think would work or does work? That's the question. At the minimum. So still quite a few people respond. I'm starting to figure out how these polls work. You can actually see what percentage of people have voted. Still a lot of people voting, a lot of people thinking about this. This is a harder question. I think we peaked there at about half the people have voted, and it appears the winner is 5 to 10 degrees Celsius. So pretty low, you know, that's kind of low. You know, if you're getting down in that range, does that have any effect? Some people have gone -5 to 0. Nobody picked below -5. You know, what is the effective temperature? Well, we have done a little bit of work on that. And what we do find is that it can be effective at a fairly wide range of temperatures, but when you start to get lower, it's not as effective. The panel on the right was done between 14 and 22 degrees Celsius with a fairly high infestation, 26% infestation of mites, crazy high level of mites. Worked great, knocked them right down, boom, pretty much down to 0. And you can see it works really, really well. At 5 to 9 degrees Celsius, we've got that 22% infestation. Not as good. It still is knocking them down. It's still kind of basically taking that mite level down to a fairly low level. You know, these were done in large rooms. So the medium large there is kind of the scale of the fumigation that we were doing. But you can see that, you know, it's not knocking it down as rapidly, not as effectively, not getting that concentration down quite as low once you're getting into that temperature of 5 to 9 degrees Celsius, which a lot of you guys put is okay, right? So temperature is affecting it. So that's something to maybe think about. Rasul Barini, an ex-graduate student that I showed in the previous slide, got a job as a research scientist working with bees in Alberta, and he did a study in conjunction with Shelley Hoover and Sam Muirhead in Alberta, which is shown on the right. And they did late fall oxalic acid applications using a number of those different devices that were in this slide that Derek provided. And you can see that it can work quite effectively in late fall. And he did his experiment in November. And the temperatures and dates in November that he applied it are kind of listed at the bottom of the slide. He applied it on the 2nd, 9th, 15th, and 22nd of November. And on those dates when he applied it, temperatures were ranging from a minimum of -1 to 0 on the first application, between -12, which is quite cold, and +3 as the maximum temperature on that day. So +3 is probably about when it was applied. -6 and +4, again, getting into that range that our previous data showed is not so great. And finally, on the last application, they did 4 applications in this experiment, was in the range of -2 to +6. As part of this project, the experiment that we did this fall looked at what happens in November in Manitoba, and you can see the results are in this panel on the left here. And what this data is showing is the percentage infestation. So we're starting with about a 3% infestation and applying it on 4 different occasions. We had 2 doses that were applied. One was just 1 application of oxalic acid with 1 gram per hive using the ProVap evaporator. The other was 4 applications. And the first application for the application that only had 1 was done on November 13th. And the temperature at that time was -13 to -5. So this is getting into that range where none of you were crazy enough to pick. I think maybe 1 or 2 of you picked temperatures that were in that range. The second application temperatures range from a min of or max of -6 to a minimum of -9. The third application, it was a little bit nicer day, -2 to -3. And the final application between -14 and -5. And you can see that under those colder temperatures, the application where we only did 1 didn't work so good at all. Very little reduction in the mite level in terms of the percent infestation if you're doing an alcohol wash. However, you look at the 4 applications, and although the percent infestation, if you kind of do the statistics, is kind of right on the borderline of statistical significance, if you take the data and you say, okay, how many of those hives, there were 20 hives in each treatment, how many of those hives would get below your spring economic threshold? So anywhere from 0% infestation to 1% infestation. And there were 14 in the treatment that had 4 kind of applications, 6 or 7 each in the ones that had 0 or 1 application. So that was actually statistically significant. The prevalence, the number of colonies that got into that safe range, even at those really low temperatures, was reduced before they go into winter. But if you remember that slide I showed you with the crazy dust everywhere, like that crazy dust is still in the hive. What's it doing? And is it still continuing to work on the bees? We often see a kind of a more long-term effect with oxalic acid. And when that dust is still in there, it's probably still continuing to work past the end of November. If you look at the mite drop, and we've been monitoring the mite drop, we're now kind of into mid-January, February, whatever month this is. I think our last samples that we took were, that we've counted the boards on, were kind of right about mid-January probably. You can see that the sticky board number there is going up until the period where those colonies went into the building. So if you look at the controls, there's not a lot of mite drop coming out onto the sticky boards. That 1% value that didn't even come out as detectable on the alcohol wash is killing mites. It's killing significant numbers of mites. It's just not killing enough to see a visible reduction in that infestation level on the adult bees. The 4 application, however, after the second application really starts to kick in, and you really start to see larger amounts of mite drop, and that is kind of reflected, if you think about it, in that alcohol wash data. So by the time you hit the end of 4 applications, you're kind of at the end of the period where, unless you're super duper crazy, you want to get those colonies into the building for winter. I'm sure most of you would not have waited until the end of November to get your colonies into winter, but we wanted to see what happens under these extreme conditions for this experiment. The kind of on the right, again, obscured from my view due to bad planning on my part, is the kind of period. So these periods are sticky boards, and those would correlate to the dates that I showed you on the previous graph. So when you're applying this stuff, it's going in there. As I mentioned, some of it's going onto the bees, some of it's probably going onto the equipment, some of it's shooting out the back end of the hive, I guess. And many of you probably are applying different amounts from what I heard at the Brandon Area Beekeepers Association. So I guess my question to you in the poll, the latest wake up poll, is when applying oxalic acid vapor, if you're doing that, how many grams would you apply per hive? So the question, 1 gram per hive, 2, 3, 4, or greater than 4. And we're getting close to the 50% part where people usually taper off with the voting. And it looks like 2 grams per hive is the answer. And that's congratulations to you, because that's what's on the provincial recommendations, 2 grams per hive. There are many people that are applying 1 gram per hive. That's kind of what we did with the package bee study. And we showed with that particular study, when you applied 1 gram or 2 gram, it didn't make any significant difference in terms of the efficacy. Some of you are applying much more, 3, sometimes 4, sometimes maybe a bit more than 4. So 4 grams per hive, that would be the dose that we showed was fairly kind of effective as a repeated application in these really low temperature conditions. But you treat kind of before winter, then you wrap them or you put them in the building, and then what happens afterwards? The mites continue to drop. So with this particular graph, it's showing you on the right here, the number of mites that continue to drop with 0, 1, or 2 oxalic acids in that period from when we ended the application to when we transferred them into the building, or they stayed kind of where they were being wrapped as a winter colony. So that application of the oxalic acid is continuing to cause mite drop during that period when the bees move into the building. Now, my next question here is a little bit trickier. I suspect the dose that I actually applied is, I might have applied 4 grams, I might have applied 2 grams, I might have applied 1 gram, but the question there was per hive. So now the question is, not all hives are 1 box or 2 boxes, right? They vary. Presumably, if you're doing it in the fall, you're either applying it to 2 boxes or 1 box. So now I want to know, I suspect, if I could guess at what device I'm using, and that my device would be applying so many grams per box. So here, we're getting pretty similar results. So people are still going 2 grams per box, or 1 gram per box, or 3 grams per box. It's kind of interesting because the provincial regulations, we were looking at them the other day, say 2 grams, but it doesn't say 2 grams per box. It doesn't say 2 grams per hive. It's just 2 grams per hive. And if you look at most of these devices, it usually says that it should be 1 gram per box. So if you're applying it to a double colony, you should probably be applying 2 grams. If you're applying it to a single colony, you should probably be applying it at a rate of 1 gram. But depending upon whether you're using one of those cannons or using Varroa or you're using a Provap, how much is actually going on to the bees? And I guess that is where you kind of see there's a few people here that have no clue, and that's probably. If I could advance the slide, that's probably the answer I would have done. That's why I kind of put, I have no clue. Like, how much is actually going on to the bees? How much is ending up on the hive equipment? How much is blowing out of the back of the hive under these different weather conditions, under these different devices? And that's a huge thing. So if we had a way of measuring that, this is one of the things I've been talking with Derek about, maybe involving the KRTP, and is trying to figure out what is actually happening in terms of what's being applied. So we can translate that into, if we produce data that says, if you apply this concentration, under these conditions, you'll get good results. And you guys can say, okay, my device is doing that. Then you can kind of match up what you need to do to calibrate that and hopefully get more consistent and reliable results with your oxalic acid treatments. The other thing is, does the wintering method make a difference? So if you're doing this indoors or outdoors, the temperatures are going to be quite different. If the oxalic dust is in the hive, or using a trickle formulation and the oxalic is still circulating around the hive, does it make a difference if it's at a warmer temperature, at a lower temperature? With Graham's work, we did study that, and he did only indoor wintering. So his colonies that showed, it looked like the oxalic was growing mites, were then put into the wintering building, and you can see that during the treatment period, the oxalic did nothing. It's no different than the control. The pre-treatment period, all these had very similar levels of mites. We actually inoculated them with the same mites, so that's not surprised. During the overwintering period, we followed them through with sticky boards throughout the entire winter. You can see that at that point, the oxalic is dropping more mites than some of the other treatments. Now that may be because the oxalic is effective, but it's still not dropping more than the controls. It's dropping more than the max or the Apivar. That may just be because there's less mites there because they were sort of killed up here in period 2. But it does appear that there is certainly some level of mite drop that's occurring in the winter. And what we found so far with the results from this study this fall is that there's way more mite drop in the indoor wintering than there is in the outdoor. It's slightly more than double. And the panel on the right-hand side shows just the total number of mites that we've counted from each of those treatments. And again, it's obscured from my view, but my memory is that the indoor had way more mites drop than the outdoor. And if you look at the indoor and outdoor zero, that's no application of oxalic, pretty much the same number of mites dropping there. With one application of oxalic, pretty much the same number of mites dropping. But when you did 4 applications of oxalic, you're getting way more mite drop in the indoor wintering. So maybe that oxalic, when you're picking up those colonies, you're moving them, you're moving them into a warmer environment. Maybe it's circulating more in that environment than in the outdoor wintering environment, and giving you a little bit more punch with your efficacy. That experiment is ongoing, and we're going to have to follow it through. Maybe when things warm up in the spring, the outdoor colonies will catch up. We don't know at this point, so we're going to have to wait till April and do the counts and see what the data shows us. So you're putting these oxalic acid applications in. Kind of showed you earlier, if you're using 1 gram or 2 grams, you're getting the same efficacy. There's not really an advantage to spending more money necessarily if you're getting the same efficacy. What about, does it have a negative impact on my colonies? Now, many of you have applied it, and I've heard anecdotal sort of incidences of people having issues with oxalic. So the poll question here, the wake-up poll again, does oxalic acid have a negative effect on my colonies? If you never use it or you don't know, you can answer that. If you think it's had an impact, you can answer that. If you think it doesn't have an impact, you can answer that way. So it's kind of tapering off now. We got up to 54% that time, so that question was apparently more interesting than my previous questions. I'm trying to go for 60, 70%. Come on, guys. The big answer, no negative impact. So a lot of you think it doesn't have a negative impact, and I suspect in a lot of cases it probably doesn't. Some people, approximately 15%, suspect there are some negative impacts, and then a lot of you, 35%, don't know. So what did we find out? First one is the experimental data from this fall, and it appears no real negative impact. Zero there is declining slightly in terms of the population size going from November 3rd to November 26th. The one treatment is stable. It's staying nice and stable. 4 applications, the population cluster size increased a little bit, and that's probably just a function of, the weather was a little bit warmer, and when you're doing cluster scoring, that cluster will expand when the weather gets warmer. So it does appear that there's, at this point in time, no negative impact associated with that 4-dose application of oxalic acid, but there's still a lot of winter left, and we don't really know what the end result will be. In the experiment we did with Paul Kozak, the result wasn't as clear. And in the experiment we did with Graham Parsons, the result was looking like there was a positive effect on population development with some of the application dates. In the experiment with Graham, we did early, mid, and late fall oxalic acid treatments, and the results were really puzzling. We actually marked age cohorts of the bees, so we knew which cohorts of the bees were being killed. And what we found out was that when you get in that critical period of the fall where that wintering bee population is being built up, you get into that kind of mid-September, those bees that emerge during that period, that's your wintering population. Those are the ones that are going to turn into your winter bees. And when we did an early oxalic application in this experiments of Graham's, it appeared to protect those bees. If you kind of look at the results, we saw no negative impact of the oxalic acid trickle formulation in this case on brood production in those colonies. The dates kind of in the early period when we applied oxalic, which is this black bar on the right, showed very similar levels of brood production as the amitraz and better than the control. When we did it in the mid-October period, again, the bar has sort of disappeared from my view, but going on memory, it didn't have a negative impact on brood at all. When you kind of look at what happened in terms of the bee population, this is oxalic applied early, and these different colors of gray bars are all the different cohorts of bees. We went in every 12 days and marked all the bees we could find in the hive. And it seems like that early oxalic acid application protected the development of those wintering bees. So one of the questions we have is how? What's it doing? Is it maybe acting as an anti-feedant and preventing Varroa from feeding? Is it maybe preventing the uptake of virus by mites or the input of virus or mites? Is it maybe affecting the sort of protein development in the bees? Is it kind of affecting the ability of the virus to replicate in bees? This is one of the questions that we want to kind of look at. We did have some, this is just the basically population size in May. You can see that the oxalic acid did way better than the formic acid treatments in the middle here. And it was as good as the amitraz. These were done with really high levels of mites, I should point out. But when you do really high levels of mites, it seems like there's a negative thing that happens with amitraz where you're better to apply late than early, which is counterintuitive, but that's kind of the results of this experiment. So the combination of amitraz and oxalic and how they interact and affect that wintering population is another really kind of fascinating question that we're kind of interested in looking at. And this shows the population size in spring. And you can see that the colonies, some of the colonies that did best were the ones that were treated just before going into winter with amitraz. They did way better than the ones that were treated earlier. And the ones that were treated early with oxalic also seemed to do better than the ones that were treated late with oxalic. This is with high levels of mites. So this is a kind of a, you're going to say that doesn't match up with what I found, but this is with really high levels of mites. And it's also, basically using a trickle formulation of oxalic acid. We kind of looked at this a little bit, and it does look like there might be something going on with the viruses. When we apply oxalic, we're seeing slightly lower levels of some viruses. We're still doing more analysis of the virus. I'm not ready to say statistically that, yeah, there's a super clear difference here. There may be a difference, particularly with Israeli acute bee paralysis virus, but this is just kind of a preliminary experiment we did, and we don't have the final word on this, but we're looking at the viruses, we're looking at the buildup of those hemolymph proteins that make the bees turn into wintering bees, and we're trying to figure out if oxalic has a protective function that we can exploit in our management. Because none of you guys are applying it early, but there may be a benefit to doing that. So what about long-term effects? Are there any long-term effects? Some people think that they thought there was some stuff happening. So what we found when we did the packaged bee fumigation, when we applied at 2 doses, is that there do seem to be some negative effects. Now this experiment, we applied it to packaged bees. We then hived the packaged bees. So all the oxalic would be on the adult bees. There'd be nothing in the colony except for what was on the adult bees. And we applied at that dose of 1 gram or 2 grams, which is the high dose. 2 grams is the high dose per package. So 2 grams for about 8,000 bees. We don't know how much dust actually went on the bees and how much went into the rest of the environment where we were doing the fumigation. But there was, when you looked at brood production, higher brood production in the ones that were hived and had no oxalic than in the ones that were hived and had oxalic. And the ones that were hived and had the high dose did a little bit worse in terms of brood production than those that didn't. If you kind of look at the colony population development after the oxalic was produced, or after those colonies were hived, and you follow them through out 60 days, you can see there's smaller population sizes. So maybe those bounce back and recover later on in the spring, but it definitely did set back that early part of colony development before you go into that exponential growth phase. And we measured the queen cells that were in those hives, and this shows the untreated, very few empty queen cells. The red bars indicate empty queen cells. With the recommended dose, we saw slightly higher numbers of empty queen cells. When we applied the 2-gram ones, we started to see cells with larvae, which are in the green bars. We started to see capped queen cells, and we started to see hatched queen cells. So that application of the higher dose to these bees and the queen in a package had an impact on the subsequent production of queens down the road. So it's disrupting something there. One of the things that we are looking at in the project with AMRO is doing those little cage studies on bees, and then we're going to send those away for all the genomic analysis, and hopefully with that we'll get a clue as to what is actually happening and getting disrupted when you get that dose of oxalic acid too high that has downstream effects on queen cell production. So that is actually what happens when you have low levels of mites. So the title there is obscured from my view, but I think the title I put on there was something like at low levels, basically there is a cost to it. When you have high levels or moderate levels, I think these were about 8% infestation of Varroa, you again see the issue, and I've got the title here at moderate levels. They may cause kind of a problem, but no pain, no gain. So you're still seeing a kind of a suppression of brood production, but the fact that you're getting that benefit from reduced mite levels means that there's really no kind of negative impact on colony growth relative to the controls. So you're knocking back perhaps the population, but at the same time, the fact that that new population has no mites means it's doing better and catching up to and probably eventually surpassing what you would find in the controls. And you got more problems with queen loss when you've got higher levels of mites. So now you get into the recommended dose and you're seeing still lots of empty queen cells being produced, but we're starting with higher levels of mites to see cells with larvae. We're starting to see closed cells, and we're starting to see hatch cells. So you've got that oxalic acid at the recommended dose now causing problems with your queens. So for those of you that answered, I suspect some things are probably happening. This is possibly what was happening or is happening inside your colonies, particularly if you have higher levels of mites. At the high dose, the 2-gram one, we're starting to see quite a bit of supersedure. We're seeing a lot more closed cells, a lot more hatch cells. So combination of high dose oxalic, and it's not incredibly high, 2 grams. That's like what a lot of you guys said you're applying. It is causing problems with supersedure. So we're looking forward to kind of basically a lot of this kind of research being carried on over the next couple of years. I do have a graduate student that's going to be working on this project that I've recruited. And as I mentioned, also we're hoping to collaborate with Derek in his new role as the Tech Transfer Research Program to find out more about what the actual doses are that are going into our colonies, so we can tie that into the biological effects that we see. The other component I'm just going to rush through really quickly because Ian wants to keep on time. He said he was afraid of me, but I'm afraid of him. So is the wintering research. And I reported on this in the last couple of meetings, and I'm not going to go into a lot of detail here, but just to tell you that we are doing it this winter. The trial's been started. We've been playing around with instead of just restricting ventilation and supplementing CO2 in the ventilation system, we are using the same techniques we've used before to put tubes in the hive. We also have meters. We found that when we do this, if we just restrict the ventilation, we can increase the concentration of carbon dioxide relative to the standard ventilation you would find in your wintering building. We've found that when we restrict ventilation, we kill more mites. That's this red bar here, 0.084% is the daily mite mortality under restricted ventilation compared to standard ventilation. And we've shown that when you restrict ventilation and increase your level of CO2, you end up with larger colonies coming out that live, and that you end up with a higher proportion of colonies living when you have high levels of mites. But this is not ready for prime time. Don't go home and turn your ventilation off in your wintering building and then accuse me because all your bees died. This is just purely high risk research we're doing right now. And it's, like I said, not ready for prime time. So I appreciate interest, but don't try it. We know that we can improve worker survival. We know we can reduce mites. How can we manipulate the ventilation? So we've got all these sensors in the rooms. We can kind of monitor remotely through the internet, actually the level of CO2 in different parts of the room. These are entrance monitors that show the parts per million CO2, and we manipulate the CO2. And we're trying right now different ways of enhancing the level of CO2 beyond just the bees ventilation. We've tried throwing pellets of dry ice into the ventilation system. What works, that creates a big spike. Not so great. It definitely increases the level of CO2, but what we're looking for is a more controlled method. And actually what works on a small scale quite well that we're gonna implement in this experiment is to actually leave the dry ice in a bag that you get it from when you pick it up, put that inside a cooler, and then you put the entire cooler inside the chamber. In this case, we've got 21 or 22 hives in there, and then the slow release happens just by the dry ice subliming through the cooler. It gets transported through the air in the entire room and seems to work quite nicely. So we're trying to actually boost the level of CO2 to a higher level to get better control and basically see how far we can push this thing in terms of manipulating the ventilation. So we actually have the cooler on a scale inside the room. You can't go in there because it's at a concentration that would be dangerous for humans. Bees are much more tolerant of CO2 than humans are. So we actually have it all sealed up. We have the cooler sitting on an electronic scale inside the room with the dry ice inside it. It's releasing over time. It releases quite nicely over an entire week period. And what you see on the right here is just the scale that we read from outside the room. So we can actually monitor the daily weight loss of dry ice and figure out exactly when it runs out. And it seems to go in a really linear fashion. It actually works amazingly well so far, fingers crossed. But and these are just the sensors that we have at different heights within the room to monitor the CO2. So this is, I guess, look forward to the results from this one. We won't have them until the end of the winter, but we have set up 2 rooms with approximately 20 or so hives each, where we're monitoring mite drop, we're monitoring, viruses, pretty much the whole shebang, looking at what happens when we supplement CO2 in the room versus the unsupplemented standard ventilation. So there's a lot of stuff going on, a lot of ideas. If you kind of look back, I like this slide. It's from 1926, Gooderam's publication of How to Winter Bees. And they did things back there not all that differently than what we're doing now. They've got colonies there that are wrapped. They looked at all kinds of different ventilation for outdoor wintering. On the right, there's guys there kind of moving their bees into a wintering cellar using a stretcher. And so I guess what I'm hoping is that, we're not doing things all that differently now than what we did in 90 years ago in 1926. So I'm hoping that we can do more than just go and do beekeeping with masks on our faces, and that as a result of this project, we're going to learn a lot of different things that we can use to enhance our management of bees and improve the winter survival. So with that, I'll open it up to questions, and if anybody has any, I'd be happy to respond. I know I've probably gone overtime as I'm known to do, but if anybody has any questions, I'd be happy to address them. Bev is waving his hand there, so I don't want to take over the role of the chair, but
okay, thanks a lot for that presentation, Rob. I think we do have time for questions. I know you started late, so Why don't we start with Bev, since he was waving madly?
You have to unmute, Bev.
The speaker on.
There we go.
There you go.
Oh,
Yeah,
good.
you're good. Go ahead, Bev.
I have an important question. When it got so stinking cold outside, my vent fans never come on for, well, a week or 10 days. And I have a CO2 monitor, and it was really quite high. Right
What's
now,
really quite high, Bev?
Is it going to hurt the bees?
What's your definition of really quite high?
Well, a couple thousand.
Yeah, that's okay. That's no problem. In the range of about 1,000 to 1,500 is considered quite normal. Quite high would be 10,000. Over 4,000 would be getting into the higher range. So those would be the temperatures, the concentration of CO2 where you would maybe be experiencing a problem. There should be, however, one fan that runs continuously in your wintering room, and there should be a standard ventilation rate of about 0.25 liters per second per hive that is continuously running no matter what the outside temperature is. So if you don't have one fan continuously running either at that rate or maybe flipping on and off with a kind of a timer to maintain that average rate over the course of a day, then I would recommend that you incorporate that.
I was getting quite afraid I was going to have to put a heat down in there to get some fresh air in, but then the weather changed and now it's less than 1,000.
Yeah.
It's more normal. Where would it become dangerous for me to be down there?
Around 4,000 is sort of the human threshold. I think at 4,000 you could be in there for several hours. I had in my talk last year the safety chart. When you're getting up around 1%, then it's getting quite dangerous, which is what we're doing. And when you get beyond that, it can, it can just knock you out. We once had a cooler with dry ice in our department that we're using for mosquito research, and the carbon dioxide is heavier than air, right? So if it's in a cooler, it just sits in there. And we had a student that reached into the cooler to pull out samples of mosquitoes and actually knocked themselves out because the CO2 in that cooler, the freezer, was so high just from storing dry ice in there. So it's something you do have to watch out. If you're transporting dry ice in a vehicle, you shouldn't ever transport it in the passenger component, because similarly, the CO2, if you don't have your ventilation fans and your windows open, can build up to levels that could be fatal.
Put you to sleep when you're driving. That's not good.
Yeah, that's absolutely, yeah, it could be quite dangerous.
Well, I was getting concerned, but then the weather warmed up and it started turning, because I like to keep it at 40 degrees Celsius, or 40 degrees
Fahrenheit. Don't put it at 40 degrees Celsius. 40 degrees Celsius is actually a temperature that can control Varroa as well. So, but it's very tricky.
Would having a higher CO2 level bother the Varroas?
That's the theory. That's the premise we're going on, is that, and we've shown that experimentally in the lab, that that does actually knock out the Varroa. We actually use CO2 as a way of collecting Varroa, and we want to collect Varroa for our experiments. We have a CO2 shake method in the lab where we take colonies that have high levels of Varroa, and we can shake them with a tube going in, blowing CO2 into this chamber, and the Varroa drop off onto a paper towel, and then we can take the bees out and the Varroa will recover, and then we can use them that way. The idea of doing it in wintering building is that if you can knock the Varroa off the bee, it hits that cold bottom board, and then it's not going to be able to get back up on the bees. So